Drosophila embryo which has a particular time stage. Gene labeled a particular image. Within the embryo, expression present. I have to mention animation up here. Animation allows you to visualizing of gene expression. Microdata. Pro is a different functions. We have different and spatial temporal situ hybridization. The naive approach is pretty hard. Image we use feature at scale and variant transfer. Cluster, we took the 15 in the 15 class to actually garbage out. What you're getting the clusters, difference in some of the clusters, but this is not really the intentionally. We want to differences or similarities within the gene. So paper called square pattern extractor. Access image pipeline patterns from Drosophila and Embryo. So over here and when pipelines, and they usually get an answer shown in B, not very normal. Secondly, you have SPEX square on the other hand outputs the pattern with embryo has more and I'll get to the and also we have done a lot of cleaning of the color where gene expression was observed. And tell you what the SPEX square pipeline looks like. We start with an image which you can see over here and track the if a particular image has a embryo, then we discuss the image. If it has multiple embryos, then we automatically determine which is the central embryo of the image. We do registration. At the registration time, we find what the anterior posterior axis of embryo is. Make sure that that's horizontal, and the dorsal ventral axis of the made vertical. Next, orientation correction is done. We define the head of the embryo and make sure that the head is to the left. Embryo should go to the top. If this was not true in the previous embryo, we flip it orientation. At the end of this, we have a standardized embryo. Size and embryo have been corrected for, and embryo exactly to subembryonic structures of the original. Hence, a pixel by pixel comparison of this comparison of subembryonic structures of the original embryo. Next, the RGB image is converted into the least in the step. You want to maximize the contrast. The regions don't have gene expression. Implementation to segment out all the noisy region pattern of the gene expression. Next, we use standard feature representation. And for quickly in our pipeline, In embryo shape extraction, we first extract the boundary of the embryo. Next, embryo boundary. Now, it could be for multiple reasons. In this particular example, when somebody was imaging, they either hit the of the embryo is not quite right. Further analysis. Boundary, we made an error, and the achieving error correction. We can take result. In the first I'm showing the top image is the embryo. It where even illumination differs in the background is light in the foreground. If there's another embryo, and this is my favorite, it turns up even if it's really hard to visualize and there is no genes present in the embryo, it still manages to do a reason. The next step of the pipeline is to do. The idea is that you want the anterior side of the embryo, which looks like this, and the posterior wall is to your right. And similarly, you want the dorsal in the middle. Now, two that we can find out what this is. Step 1, where use the heuristic and that the dorsal side is and build up some features and correct for it. Analysis. There might be gene at the same time. Join. If you have two embryos, I have expressions embryo, but they belong to the same gene expression, then that tells you one of your embryos, and we have an inference algorithm that helps you determine to correct for its orientation. Thus, we get better orientation results. The original image, the AP axis. In the second one, horizontal axis. In the third one, orientation of the original overall. And so, which are the bottom. And as you can see, that's really nice. So far, I talked about how the embryo standardization is done. Next, I'm going to talk about how gene expression pattern extraction is done. How we convert the RGB images into simple RGB into grayscale conversion, because we want to maximize the contrast between the places that have gene expression, and the details how we do this conversion are time to go into it. So what happens is, even though if, once we've converted the scenes, the input image that we have, which was the grayscale image in this row, will be noisy. The last image over here is really, really dark blue, and the at the imaging time. So we put a marker field, which is a graphical model, to automatically segment the image into the places that had gene expression and the places that did not. So here's an input image. It's a grayscale image. We're going to construct a field that is specific to this particular image. We do this in a unsupervised way, where the structure as well as the parameters of the MRF are learned in an unsupervised way. So first, the image is over-segmented, and connectivity information of the over-segmented regions is used to structure of the Markov random field. This is a pretty standard approximation in computer vision. Next, we use the parameter of random field by simply running X-program of the model. Now that we have the we use belief propagation to do inference. And so the Markov random field tells you what parts of it, and And that's for background. The background is set to 0, 2. The final result that the original image. And in the bottom row, as you can see, SPEX gives extremely precise pattern, and they capture parts of the original independent size orientation of the embryo, and it's of error correction. You don't get much noise in the final outputs in further analysis. So final expression pattern that is converted into features by using the SIFT. Dimensionality reduction is used to convert it into a reasonable sized data set by further algorithms. About the pipeline of SPEX. And the next question is of genome project lateral view from the 13 to 16 time spent. We have 2,689 genes, and at the end of processing, our pipeline extracts 19,011 genes. We're going to use the feature we've extracted and apply them to two distinct tasks. Imaging. You have a bunch of images created, and with labeling where was the labeled as expression repeats ectoderm and so on. Do such annotations by ourselves. So since this is and a separate from its own right, we simplify only annotate images 1 of 10 possible, and this becomes now a classifier. I'm going to run it with a few systems. The first list classifier that we think of, which is support vector machine. System 2 are discussed in the paper, but previous literature that's reported. System 2 is specialization, which is extremely sophisticated and predictions between the application terms themselves. And regularization classifier to see what kind of results you get. Here are the results. The first two blue bars where you localization and support vector machines, and those are the other two systems. Reporting results on two separate metrics: mean accuracy, you get correct in your estimates, and the higher the better. F1 of precision recall, and again the higher the better. Both of these we observe that SPEX is capable of doing significant, and results are with the accuracy, which is 6.3 e raised to minus 10 raised to minus 6, 10 raised to minus 5. So both of them are significant Bruce lines. The next task that class 3, the original problem that we started was that we so one way of doing that is to say that we want a cluster similar to a single cluster. So if we are clustering based on spatial similarity, it means that we also get a similarity. That's the key question that we're trying to answer. Functional similarity by using standard Go ontology. So I'm again going to emphasize it's a problem. This is the plot I showed you with the mean images from every class. This is what system 1 gives. This is actually a much better some embryo. These two are completely noisy. They have reasonably features, and you don't, and the results don't look very nice. This SPEX produces, as you can see, that each is the mean of images assigned to a cluster. The mean of around 100 images. And even then, extremely visually coherent. You can clear a visual pattern that this is corresponding to, and the other systems we've seen so far. Some examples of images put in the same cluster, and not surprisingly, they look very similar to each other. Here, images put in the same cluster by SPEX look very coherent to each other. We found that 9 terms, but I'm related to love bristle morphology active only. Surprising clustered dependent of how many clusters we picked. For enrichment of for a variety of clusters all up to 100 of these clusters significant at equals. We find that there are no clusters that signal the remaining two systems. We do the kind of in network analysis, we take in this particular network from a particular this is from cluster network networks, and again the other nice thing is in cluster the embryos separately. The idea is we found we similar example the region behind and we have images. The advantage they can support on end with acknowledgments research.
Of the embryos.
Use gradient. The front of them should be pretty to get and erosion
Concise's measure. I mean, it's obvious.
So, using multiple data set. The best result.
The images you show.
Well, you usually noise.
Right, and
Actually was turned off. You
have a cut.
This particular of should be concerned about.
I think there needs to be RNA sustain.
Okay.
And so the classic one is not the same. When you tell me, how are you distinguishing?
Only one data. I can say that, oh, go look at some other. Only information I have is this particular information.
Data, yes?
Yes.
These data are RNA. You're trying to relate an RNA hybridization experiment.
But so I think that you're kind of assuming that those are some assumptions that field makes. I don't see how you can
definitive not here.
They're exactly not most.
Let's talk about this offline.
Right. Yeah. Construct, and I think it's a stand. Forgive me, I can't remember. I think
These are all the same promoter.
That's right. That's right. Yeah. I apologize. I have my head in the details. I wasn't here who then that's Pavel Tomacek, who's Pavel. Come back, introduce him, and you can get very definitive answer. He was one of the.
Okay. Yes, thank you. You have a question?
This is a very quick question. So, when using the embryo, the subgene clustering analysis, divide the embryo into triangles, a lot of triangles. It seems that way that you from the derive the functional subgroup.
So actually, that is ongoing the triangle method as well. We do have some results that should, but directly use the gene expression patterns to do clustering. But those are much harder to analyze for us as computational. This is a finding and help us analyze those results. Okay, thank you.
Okay, and I also worked against the data. It's definitely mRNA.
Okay.